my great pleasure to introduce the first speaker, Dr. Marie Harrington. And so she's been honored today, given her contribution as a chronobiologist to Canadian chronobiology and as a researcher working abroad. So Marie is now a professor, a Tippett professor in life science at Smith College in Northampton, Massachusetts. And she has been developing over the years methods for measurements of organ-specific circadian gene expression rhythms in freely behaving mice. So she has published a textbook also recently entitled The Design of Experiments in Neuroscience. And she has been receiving notably the Sherard Prize for Distinguished Teaching. So Mary is currently the editor-in-chief of the Journal of Biological Rhythms, and she has been really tremendously contributing to undergraduate teaching at her institution. She was sharing this information with us yesterday, in particular during lunchtime. So it's really a great pleasure to have you here, Mary, and it's an honor for me to give you this little gift. On behalf of the Canadian Society for Chronobiology.
You have those options, yes, the microphone and
It's okay?
yeah, you can install it. So, okay.
Thank you. Thank you very much for this recognition. I'm really delighted and surprised, and it's a pleasure to be here and to get to know all of you and hear about your work. So I'll tell you about some of my recent work, and I'm going to start with an outline, but also I invite you to take a shot of this and connect to the poll. So if you have questions during the talk, we often don't have time for questions, or maybe you're a little shy about me putting your question out publicly, so please post your question on this poll. It will collect your email, and I can get back to you, or we can connect with the meeting. So first I'm going to talk overall. I'm going to focus on peripheral clocks and tell you a little bit about what I know in general about those clocks. I'm going to describe a new method, then tell you about an old technique, and some new results combining those two. So let's get started. So when I started as a graduate student, this is how we thought about the circadian system. It was a hierarchy. The SCN got input. It was the pacemaker of the peripheral oscillators. So that's the way I would draw it. The SCN's getting light input. We used to call it the master clock and the other oscillators the slave oscillators, and I'm just pointing you to a recent editorial in JBR written by a graduate student pointing out how maybe this master-slave language is language we should move away from. And I invite all the students in the audience to think about what you might want to contribute to our journal. And you can look at the argument for this change in language. As we think about what language should we use to describe this clock system, I think we still need better terminology. And so yesterday we talked about should we talk about it as an engine with interconnected parts, or an assembly line, or an orchestra? But I don't know what this figure is showing, a system of pulleys maybe. I think we're still a little confused, and some people have suggested a jazz band, which I think is actually a really nice metaphor, because as you'll see, sometimes some parts can just take off on their own and do their own thing, separate from the director and the SCN. So this is a work in progress, trying to figure out the best metaphor. I think it's important because metaphors shape our thinking. So how can we begin to study these peripheral oscillators? Now I'm going to tell you about our new method. It grew from early work where Joe Takahashi's lab developed an animal, the Perlock animal, where you could track the period 2 gene through this fusion protein between period 2 protein and luciferase, firefly luciferase. And shown on the slide is the in vitro lumicycle equipment that many of us used, where you could just slot in little culture dishes and measure bioluminescence coming from different bits of tissue from this mouse. And as you see in the center, the SCN showed showed persistent rhythms in bioluminescence when cultured. And interestingly, the same paper showed that the liver and the lungs, so some of the peripheral oscillators, could, for a month, generate circadian rhythms in culture isolated from the SEN, suggesting that we're missing some important aspect. At that time, it was very, very surprising, and it still surprises me, that peripheral tissues can function so well as independent oscillators. To follow up on that work, I did some work with primary cultures of liver hepatocytes, and we could see the bioluminescence in these primary cultures showing a rhythm, and that allowed us to then ask questions about how are these cells coupled. So we assume if each cell is an independent oscillator and they're maintaining an overall rhythm in culture, the cells must be coupled somehow, exchanging circadian information. And so we did experiments. The strongest experiment in this paper was when we combined different percentages of cells with different circadian periods. So in this case, we used a Cry2 knockout, and the circadian period of those cells is closer to 29 hours. Wild type cells more like 23 and a half. And so if you made a culture, say only one of these cell types is bioluminescent, but you start adding like 75% Cry2 knockout cells, do you see a change in the period? You would if the cells were coupled, and we didn't. So that would indicate there's not strong coupling, at least in our cultures. We did see some evidence for weak local coupling. Cells that were near each other were more likely to be similar in circadian period than cells that were randomly selected across the dish. So this is perplexing to me, and I wanted to study this further. But I needed a new method. I needed a way to get away from some of this artificiality. I was worried about the cell culture. This was pre-organoids. Maybe now we would have better luck using liver organoids. But I wanted to find a new method. And another reason I wanted to find a new method is that I spent many years doing what I call in vivo, in vitro studies, where we manipulate an animal in vivo and then quickly dissect the animal, put tissues in vitro, and look at circadian phase. And using, again, that per-luck animal. And in this study, we sort of killed it for ourselves because we showed that after circadian disruption, now the circadian phase of the tissue in culture was completely set by the time of dissection. And that wasn't true in control animals, but it was definitely true following the circadian disruption, in this case through LD1010. A surprising result for us, and we weren't completely sure of it until, I always like that when you see someone else finds the same result. And so another lab using a very different form of circadian disruption saw the same result. So I would say we're pretty sure now that after circadian disruption, one effect of circadian disruption is that our tissues are more sensitive to the effects of dissection. So what's the new method? This started with a paper published from Schibler's lab. Saini was the first author in 2013. And they describe in that paper the way they developed this new method for measuring bioluminescence coming from a freely behaving animal. And they proposed and patented this highly engineered, Swiss engineered equipment for measuring bioluminescence from a freely behaving animal. Lovely, priced way beyond my ability to bring this into my little lab. And so I started thinking along those lines and started working with David Furster, who is the person who created the in vitro lumicycle. And we sort of pulled out the guts of some of that and made it so that you could just slip in the animal's cage, and the photomultiplier tubes on the top would now, instead of counting bioluminescence from the tissue culture dishes, they're counting bioluminescence coming from the animal. And if you house an animal, a per-luck animal, in this cage and provide luciferin, even just in the drinking water, you can see the rhythms of bioluminescence coming from the animal. So you can now measure, in this case, all peripheral clocks. I don't think the light's getting out of the brain, so I think this is really just all clocks of the animal are synchronized enough so that you can see an overall rhythm from this animal. It's a noisy rhythm. I mean, the animal's moving around its cage. When it's noisy, but when you look, in this case, I'm really just showing you this to show you the raw data and the little data inset. It's noisy data, but here we have an N of 3 versus 4, and we can see a sex difference. So this is also a new finding, that there's a sex difference between males and female mice. The females have a slightly earlier phase in this perluck rhythm and are less variable than the males. And so these studies indicate we have a new technique, and it's one that we can use to make new discoveries. But I don't really want to study all the peripheral clocks of the whole animal at once. What we really want is a way to select one tissue and just study that, like the liver. How do we do that? And here I really benefited that in parallel, Robert Dahlman and Dave Weaver were leading a study that became Sierra Smith's PhD thesis, where they developed a new animal using the DBP circadian clock gene, linking it to Firefly luciferase in a way that you can direct that to specific cells. And I also appreciate that they decided to publish this in the Journal of Biological Rhythms. We're all supporters of this journal. It's a flagship journal. It's the Society for Research on Biological Rhythms. And I invite all of you to contribute some of your best work to this journal, which will keep it very strong. So using this DBP reporter mouse, they showed that if you combine that, say, with albumin Cree, which directs the reporter to the liver, you can show through, in this case, IVUS imaging, that there's light coming out of the liver of the animal in a rhythmic manner. So that helped us, this imaging helped us be confident of where the light's coming from. Because with the technique I'm developing, it's not imaging. You're just counting light coming out of the animal. So you need to combine it with these sort of techniques. But when we move to looking in our lumicycle in vivo and using these liver-directed reporter animals, we could see that DBP was an excellent choice because it's not expressed at all, and then suddenly it's expressed. So DBP has an extremely strong, almost a square wave of expression in the liver. And the other thing I really liked about it is the DBP expression increases well before the animal becomes high in locomotor activity. So we could feel more confident that we're separating what we're measuring as bioluminescence from the animal separate from the locomotor activity. And we did a few studies just to characterize this, to show what we pretty much thought we would show, that following jet lag, the liver was slower to shift than locomotor activity. This verifies prior results. That if you fed animals during the daytime and then release them into darkness in our in vivo lumicycle, you can measure that the liver is reset to a new phase. So these are expected results. And so now I'm going to go to tell you briefly about the way we applied an old technique. This technique is doing SEN ablation. So it's stereotaxic surgery. You can put down 2 electrodes. You can ablate the SEN. It's very difficult to do, in fact. You have to be a pretty good surgeon. I was lucky that Chen-Yu Lo joined my lab at that time and was able to master this technique really rapidly. And we're able to have animals in the lab with SEN lesions. The SEN lesion is a technique that can make the animal behaviorally arrhythmic. So now we have an animal where the SEN is arrhythmic and no genetic changes to the animal, and we can begin to study the peripheral clocks in this setting. So I'm going to show you new results. This is, we haven't published anything on this, and as you'll see, we have a lot to learn still. But I think we're poised to do some fun experiments. So when you look at the liver in an SEN ablated animal, it doesn't stop. It's rhythmic. It keeps going. It changes a little in amplitude this is over a month. So over more than a month, SEN ablated animal who's behaviorally arrhythmic, the liver is perfectly rhythmic. I know that I should have known this from the paper in 2004, but I didn't believe this would happen. I was really surprised that an animal who looks behaviorally so arrhythmic could have such a strong rhythm. And there's a lot we don't know. What's generating that rhythm? There's a lot of questions. If you look at that same animal in actigraph format, you can see that rhythm is clearly circadian. It has a period that's different from 24, which is a relief because it's not driven by something in our building. It shows changes in period. It shows abrupt phase changes. So we can study. We can't see anything in our setting that can explain these things. The locomotor activity is clearly arrhythmic. We've looked at drinking, that's also arrhythmic. We've looked at feeding, that's also arrhythmic. So the animal is behaviorally arrhythmic, but the liver is rhythmic. Actually, another paper from Schibler's lab, Cinderell, had published data that I could take, because they published the data file with their paper, I could take their data and analyze it the same way I've analyzed my data, and you can see that they have very similar results. They're using adenovirus vector to deliver a different reporter, another about reporter linked to luciferase in animals that have SCN lesions. So now our field has an N of 2, and it looks like maybe this is trustworthy, and we can talk about what are the properties of the liver clock in an animal that doesn't have an SCN. So I'm not sure about this hierarchy idea. I'm thinking, there's the liver has a lot of capabilities. I want to study that more. It might be coming from other clocks, so there could clearly be inputs from other oscillators in the animal. It's going to be interesting to study. Sometimes I get interested in other topics, so my eyes were taken over to new work also by Ethan Burr. Very interesting work about the clocks that surround us in our skin. And so I'd never thought about that we are covered in circadian clocks, and what Ethan showed in this really wonderful paper is that those clocks are photoentrainable, separate from the SCN. So he has a case where the SCN is free running, the SCN is intact, but just not getting retinal input, and the clocks in the skin are entrained to the light dark cycle in the whole animal. So I was like, I need to study this, which means that we need to make a skin reporter animal and then look to see what are the properties of light entrainment. And now we're thinking, okay, if light comes into one of the peripheral clocks, might it then transmit light information to other peripheral clocks? It's possible. So just to give you a taste of what we're doing, we're measuring rhythms from an SCN intact animal, and we can measure rhythms in bioluminescence. When we look at SCN ablated animals, we do see rhythms in bioluminescence, but after about 2 weeks, they damp. And so they're not as persistent as the liver. How do we get them started again? We use dexamethasone in some experiments. Many of you would do that too. And then, I don't know, just weird experiment, we gave the animal a companion. So here's an animal, SCN ablated animal, skin reporter. He's not showing a rhythm in bioluminescence until he gets a companion, he or she. We've done this with males and females. And the companion is not bioluminescent, but it induces a rhythm of bioluminescence in the skin, which opens a whole bunch of questions about, is touch or temperature or what, synchronizing skin oscillators. So I feel like we're going to have fun examining that and asking a lot of questions about skin rhythms. The work that I do is done by undergraduates training in my lab, so many students have contributed to this work, as well as a longtime technician, Penny Molyneux. And as you can tell, I collaborate freely, so I've highlight here some of my main collaborators with the DBP reporter and the skin work. And Tanya Lysy has been a key collaborator for me for many, many years, contributing a lot of the analysis, and I really couldn't be here without her work. She's recently passed away due to cancer, and it's a big loss. So, thank you.
Thank you so much, Mary. There's already a question, Jennifer?
Jen?
Mary, that's beautiful as always. You're asking questions about how these all interconnect, and whether the liver is running by itself or something's feeding in. We know the liver is very sensitive to food, time of day it's eating. Can you or have you done experiments where you give a bolus of food to see that that's what's resetting the rhythm so that you can see it's kind of independent? Or have you looked at other cues that might be directly resetting the liver to know whether or not it's liver driven versus other driven?
Yeah, we haven't done that yet, but I feel like the giving food is going to be an easy experiment. The question of what is driving it is a lot harder. So I'd love to talk with people about strategies for quickly ruling out peripheral clocks. I mean, from Frank, I realized that we don't have to worry about melatonin because these animals don't make melatonin, but there are other possibilities that might be driving this rhythm. So I'm not starting from the idea that it's coming from the liver. I'm starting from the idea maybe we first rule out other sources. Sachin?
It's beautiful work, but it's also, if you look at mice that are continuously fasted for 36 hours or 48 hours, you still see a very strong liver clot. And there are many experiments with high fat diet, if you eat in every 1 hour, there's liver clot. What is actually missing, what misses is the coupling of the liver clot to its output that are important for metabolism. So, since DBP is a direct target of proteins, maybe something outside the core clot component, for example, CYP7A1, which is strongly modulated by clot, but it actually goes flat in continuously fasted mice, some of the clot output that are not direct target. Those might tell us what is the importance of the CN, because the CN, dry feeding fasting that in turn couples the clot to its output.
That's interesting. And you know, in many cases, we're killing animals at different times to measure rhythms. But in this case, when we can measure the DBP rhythm, we can then select animals at times when the DBP is up or down. Because I think you're getting at a question of that I'm interested in is like there's a rhythm in DBP, but is there a rhythm in liver function, and in what way? How is liver function altered? But I think with this animal, we can at least know where is the rhythm in the liver, which otherwise you wouldn't be able to know. Yeah.
Well, Frank, and after Florian, and I think we're going to continue the discussion after Frank.
Mary, thank you so much. Really exciting what you're doing. I was curious to see your pattern of activity of DBP, and so it almost seemed like there was relative coordination, meaning as if there were two oscillations going on that were interacting. I don't know if you can pull up the slide, but so yeah, do you have, so because it seems that the liver clock period is shorter than 24 hours, but there also seems to be a still remaining 24-hour component running through as well.
Yeah.
So have you considered relative coordination, and are these animals in DD or LD?
So I like that question. The animals are in DD, but they are checked every day.
Right.
The students call it random times, but of course it's during our activity period. And so, and Giselle Oda in Brazil has shown that that would be enough to entrain animals if you just did random times within a 12-hour window. So I'm a little nervous about anything 24-hour running through this. Also, there's a rhythm in the building, right? There's building noise.
Right.
So I appreciate that question, but I was, and I would expect you would pull out two components. The strongest component in this actogram, it looks, when you analyze the rhythmicity, there's one main peak, and that's the longer period one, but I appreciate that. Yeah, yeah, yeah.
Yeah, fascinating work, and I'm glad that you have this slide on, and I want to draw your attention to the lower panel, and the left one is the lesion animals pattern. And as you know, I have an interest in ultradian rhythms, and when you take a look at the ultradian rhythms in the middle section, they are actually in phase with the liver. Do you see that?
I see that, yeah, yeah.
Which is fascinating. I mean, just, you know, starting point to think about who's talking to whom, but just interesting.
Yeah, yeah,
fascinating. Yeah. Only your study could show that. You know, you have the tool at hand to understand it.
Yeah, yeah, yeah. Yeah, there's a tiniest little 24-hour rhythm in Central's actogram, but it's also, the downside of an SCN lesion animal is every animal's different. You know, every lesion's a little different. And so it's, that's the downside of this approach. And so we will have some animals with partial lesions that have a little bit of rhythmicity left.
Maybe you have to cross it because people want it. It's a hassle.
I started that way, but I'm convinced there's no good genetic way to do this yet. Every time I meet Joe Takahashi, I say, give us a way. Knock out the clock in the SCN. Only.
Only, okay.
You know? But so far we don't have a way to do that. What's he doing with his time?
Please join me in thanking Mary again.